and welcome to my presentation on stem cells. My name is Destiny Gibson and I am going to take you on a journey. This is a table of contents. You can pause it and write these down if you just want to be ready to take some notes. But let's get started. So firstly, what is a stem cell and what does it do? Stem cells are what we call undifferentiated cells. This means that they are not specialized for certain parts of the body, and there are two things that can really happen from here. Stem cells can either multiply through a process called self-renewal and create more stem cells, or they can differentiate into specialized cells that have a specific purpose in the body. Before differentiation, stem cells do not have specialized jobs, but they do have access to a large library of genes, giving them the potential to become a wide variety of cells that do. Now, the ability that stem cells have that allows them to differentiate into different types of cells is called potency. Stem cells have different levels of potency. Totipotent stem cells can differentiate into any type of human cell. After fertilization, the zygote and the cells from the earliest cycles of cell division are totipotent stem cells, but no other stem cells are totipotent. Then you have pluripotent stem cells. Pluripotent stem cells can differentiate into most types of human cells, but not all. These stem cells are found in embryos and induced pluripotent stem cells, which we'll talk about more in the next slide. Finally, multipotent stem cells can only differentiate into a few types of cells. They are restricted to a certain type of tissue. These stem cells are found in many adult tissues, including bone, cartilage, blood, muscle, fat, and neural tissue. Now, let's circle back a bit, and I'll tell you a little more about pluripotent stem cells. There are two types of pluripotent stem cells: embryonic stem cells and induced pluripotent stem cells. Embryonic stem cells are abbreviated as ESCs or HESCs, while induced pluripotent stem cells are abbreviated as iPSCs or HIPSCs. ESCs are grown in laboratories where a donor egg is fertilized with donor sperm through in vitro fertilization. On the other hand, iPSCs are created by genetically modifying adult somatic cells. Transcription factors, small molecules, or microRNAs can induce pluripotency in adult stem cells that are normally not pluripotent. These genetic modifications are created by using retroviral transfection, transposons, plasmids, mRNAs, or proteins. Just give it a second. My computer is... There we go. Now, one major thing stem cells are currently used for is bone marrow transplants. Bone marrow stem cells, or hematopoietic stem cells, are multipotent, and if you think back to that second slide, you'll remember that this means they are adult stem cells that are restricted to a certain type of tissue. Bone marrow stem cells can differentiate into white blood cells, red blood cells, and platelets. Bone marrow transplants can transfer the stem cells of a healthy adult to a diseased patient. They are used to treat diseases such as leukemia, lymphoma, and inherited blood disorders such as anemia. Bone marrow transplants are often life-saving, but they can also be life-ending. One of the risk factors involved is death due to tissue rejection or infection. Now, next we're going to talk about some of the more recent major innovations involving stem cells. Very small embryonic-like stem cells, or VSELs, were first discovered in 2006. They are pluripotent stem cells that are found in adults instead of embryos. VSELs are very rare and difficult to isolate, but can differentiate into a wide variety of tissues. They are a promising technology because they can replace the need for embryonic stem cells and work as an alternative to induced pluripotent stem cells. Another recent innovation is the organoid. Organoids are miniature organs grown in vitro from stem cells. The hope is that one day they will be able to use a patient's stem cells to grow new organs to replace their failing ones. One major example of this is the miniature heart. In 2019, in Israel, a group of researchers at Tel Aviv University used personalized bioink created from stem cells from a patient to 3D print a tiny human heart. It wasn't functional, and in the past, people had been able to 3D print miniature hearts too, but in this case, they were able to print specific cells and blood vessels, which was something they weren't able to do in the past. And best of all, because the organ was created from the cells of the patient in need of a transplant, they found that this new technology has the potential capability to eliminate the need for immunosuppressive drugs to prevent rejection. And then earlier this year, 2020, at Michigan State University, it was revealed that they made an even bigger stride in this technology. They were able to print a functioning miniature version of a human heart. Next, and finally, we'll talk about human limb regeneration. Have you ever seen one of those little salamanders? The ones that can lose their tails and grow them back? Well, the research on human limb regeneration is based on this ability. Losing a limb for a human can be a life-threatening ordeal. Right now, this research is in the introductory stage. They are examining evolutionary lineages and trying to figure out if regeneration in humans is plausible. There have been cases of small children regrowing the tips of their fingers, but can it be done on a bigger scale? Well, the answer is a very enthusiastic yes. And they hope to see strides in this research in the near future using stem cells to tailor the limb specifically to the amputees. Finally, we'll talk about why we need VSELs and iPSCs to replace ESCs. Embryonic stem cells have a major ethical component attached to them that has caused a lot of controversy. Now, the main issue ethically when talking about embryonic stem cells comes from a religious standpoint, mostly the Catholic Church. They believe that the moral status of the embryo should be taken into consideration. It's based on the belief that as soon as the egg is fertilized, the embryo has a right to life, or the right to be born. So the use of embryonic stem cells in research, which are harvested from embryos created in labs, is believed to essentially be like killing babies. The controversy is much like the debate about abortion, and if abortion is ethical. The question is, when does an embryo become a person? When it's fertilized? When it has a heartbeat? When it's born? That's the end.